All right, good evening and welcome to the October installment of VIMS's After Hour Lecture Series, our last of 2020. My name is Candace Vinson and I am the Outreach and Events Coordinator at the Virginia Institute of Marine Science. I'd like to take a moment and just tell all of our sponsors, thank you so much for helping us make all of these events happen and allowing us to still bring this information to you virtually when we cannot join in person. So let's get into tonight's presentations. Bruce Collette is an ichthyologist with 60 years experience with the Bureau of Commercial Fisheries, now known as the National Marine Fisheries Service of NOAA, and is a research with the Smithsonian National Museum of Natural History in Washington, D.C., and the chair of the Tuna and Billfish Specialist Group of the International Union for the Conservation of Nature. He is a leader in the field of marine ichthyology and conservation and has served on a total of 17 VIMS committees for VIMS graduate students. Both John Graves is the Chancellor Professor of Marine Science at the Virginia Institute of Marine Science and an internationally recognized expert on the biology, fisheries, and management of tunas and billfishes. He has served as chair of the U.S. Advisory Committee to the International Commission for the Conservation of the Atlantic Tunas for the past 25 years and been a member of the National Marine Fisheries Service's Highly Migratory Species Advisory Panel since its inception in the late 1990s. Val Kells is a widely recognized freelance marine science illustrator with over 36 years of professional experience. Her work has appeared in over 30 public aquariums, museums, and nature centers. She has also recently co-authored and illustrated 5 notable field guides of coastal and freshwater fishes. So now I'd like to welcome Bruce Collette first, and then he will be followed by John and Val. Bruce, take it away.
Thanks for your introduction and greetings. This evening, we will tell you about our book, and there's the cover of the book on the screen now, and its contents and some of the history leading up to producing the book. Then John Graves will tell you a little about billfishes, and Val will describe how she makes her lovely illustrations, some of which are shown here on the cover of our book. We plan to finish with enough time for questions. Our project began when the three of us met with Vince Burke, our original Johns Hopkins editor, on November 29th, 2012. So that's 8 years ago. Who do we write this book for? For sports folk who pursue sailfish, marlins, yellowfin, and bluefin tunas at great effort and expense. For commercial fisher folk who harvest vast quantities of mackerels, bonitos, and tunas to feed the people of the world. For fishery biologists, whose task is to efficiently manage populations of these fishes for the benefit of people without endangering the stocks. For conservationists, worried that species like the bluefin tunas might be fished to extinction. For biologists interested in the anatomy and physiology of the great tunas that enables them to maintain body temperatures above the temperature of the water they live in, unlike the vast majority of cold-blooded fishes. For biologists, tracking the migrations of Atlantic and Pacific bluefin tuna across oceans to feed and then to return to the waters where they were hatched. But why tunas and billfishes? These two groups of fishes are not closely related, as shown by their morphology and genetics. But both groups contain large, beautiful epipelagic fishes of great interest to sports fisher folk, commercial fishers, and naturalists. Many species of tunas and mackerels are the basis of large commercial fisheries worth billions of dollars. Tuna have been the 4th most valuable globally tracked fishery product. The first big Pacific bluefin caught in 2020 was auctioned off in the main fish market in Tokyo for $3 million. $3 million for one fish. But that was at an auction similar to those held in the US by restaurants for a prize steer. Normal prices might only be $5 or $6 per pound, or maybe up to $20 for prime bluefin. Fresh mackerel, canned or grilled tuna, sashimi, all delicious components of our diet. Sports fishermen spend many days at sea on expensive boats and large sums of money in pursuit of marlins, sailfish, swordfish, yellowfin, and bluefin tuna. Revisions of most groups of tunas and billfishes, plus all the information gathered for the species accounts in the 2010 Red List of Endangered Species, published by the IUCN, the International Union for the Conservation of Nature, served as the background for our book. We spent 6 years writing and revising accounts for the 61 species of tunas, mackerels, and billfishes. Starting with the less well-known species, I searched the literature and drafted accounts, species by species. I tried to find useful references in as many different languages as possible to make the information available to a wide audience. So we have references in at least 9 languages, including important papers in Farsi and Indonesian. However, the list of references became too large to comfortably fit in the book, so Johns Hopkins University Press agreed to make the entire list of over 100 pages of references available online. John then reviewed the draft accounts, adding fisheries information and updating the biology and threat status section. Val then created layouts for each species account, adding her illustrations and editing the text to fit into our format. All 20 components of each species are shown here on page 13 of our book. After some personal history, I will show you the layout for a sample species of Spanish mackerel. People frequently ask me, how did you get interested in tunas? I was a graduate student at Cornell University studying the taxonomy of a group of small freshwater fishes called darters. Robert Gibbs, who had been a graduate student at Cornell, was studying large pelagic fishes such as tunas and dolphin fishes at the Woods Hole Oceanographic Institution. Bob hired me as a research assistant in the summer of 1957. My first introduction to tunas came from our participation on 2 cruises of the old Bureau of Commercial Fisheries vessel, the Delaware, using Japanese longline gear to catch tunas. This was the first use of this gear in the Western Atlantic. Tunas were abundant, and boy did we catch them. Bluefin, big eye, yellowfin, and albacore. And we gathered all the data we could on them: color, weight, stomach contents, parasites, everything we could think of. After graduating with my doctoral degree from Cornell in 1960, I was hired by the Bureau of Commercial Fisheries, now known as National Marine Fisheries Service, at the Ichthyological Laboratory based in the National Museum of Natural History in Washington, D.C. Jack Marr, then with Fish and Wildlife Service's Pacific Oceanic Fisheries Investigation, asked me to straighten out the taxonomy of tunas, figure out how many species of tunas there were, what their proper names were, and do it well enough that the damn names didn't keep changing. At about that time, as an example of the problem, yellowfin tuna in Hawaii were called Neothunus macropterus, while they were called Thunus albicares in the Western Atlantic. Not just different species, but different genera. This meant that researchers in the Western Atlantic didn't use biological information from Hawaii because they thought it was coming from tunas in a different genus. Gibbs and I spent 6 years answering the questions Jack Marr had raised, concluding in 1967 that there were 7 species of large tunas: worldwide albacore, yellowfin, and big eye, 2 species of bluefins, Western Atlantic blackfin, and Indo-West Pacific longtail. And our morphological analysis showed there was only 1 worldwide species of yellowfin, later confirmed by genetic data. Why did it take us more than 6 years to sort out the species of the great tunas in the genus Thunus? Tunas and billfishes are large predators that swim rapidly through the water in pursuit of their prey. So they must be streamlined for this, like airplanes, cars, and boats. While the external morphology of tunas is similar, the internal anatomy has evolved different modifications in each species. This means that it's necessary to dissect tunas to find differences in their circulatory system, swim bladder, liver, and skeleton. We did some dissections at sea, but relied mainly on bringing specimens of these large fishes back to the lab or having them shipped to us frozen. Here's a figure of the skull of a dogtooth tuna to show how numerous the bones are in a fish skull. We compared all these bones, specimen by specimen, species by species, in order to figure out what their classification was. Higher tunas are warm-blooded. Their body temperature is significantly higher than the water in which they live. How do they do this? They have evolved a countercurrent heater system so that the oxygen-rich blood in blue coming from the gills flows along the body next to blood vessels that are warmed by the extensive red muscles. Here's the blue vessels coming this way, and then you see there's red vessels parallel to it. And as you see down here, the red and blue vessels are right next to each other. So this means that this oxygen-rich, cool blood flowing this way is next to the red, which is the warm blood coming from the muscles. So that results in the heat staying right within the body, and the fishes are warmer. In 1983, FAO published our summary of the species, of all the species of tunas and mackerels, with identification keys, drawings of each species, distribution maps, and summaries of biological information. This publication and a parallel publication by our Japanese colleague Izumi Nakamura on the billfishes have served as major sources of information for many years, but have become outdated and needed updating. With the systematics of the large tunas worked out by Bob and myself, my colleagues went on to sort out the names and number of species in other parts of the family. I started with the bonitos and relatives, which I revised in 1975 with Labish Chow, who had been a PhD student at VIMS. Then the Spanish mackerels, Scombromoras, including discovery of 2 totally undescribed species. Then the double-line mackerels. Here's the 2 species of double-line mackerels. The one on the bottom grows much bigger than the one on the top. The one on the top is widespread throughout the Indo-West Pacific. The one on the bottom with these little black spots along the ventral surface is restricted to Australia. Then the frigate tunas, and here's the 2 species of frigate tunas. And maybe you can see the 2 species differ in the width of the corselet, that area of here. You see, and here the corselet extends way back here. Following that, we worked on the mackerels of the genus Scomber with John Graves and Dan Scholes, another VIMS graduate student, in 1998. So here's a summary of the species level changes that we made within the Scomberdae. Recognize the yellowfin tuna as a single circumglobal species. Recognize the southern bluefin tuna separate from northern bluefin tunas. Describe new species of Spanish mackerel from Latin America, and we'll come back to that one. And another one from Australia. The two new subspecies of frigate tunas, the Pacific chub mackerel recognized as a separate species, and the Pacific bluefin separated from the Atlantic bluefin as a separate species. A story about Spanish mackerels shows why it is important to know the identification of the species you're managing. That's really the first step. U.S. fishery biologists were trying to manage populations of the Atlantic Spanish mackerel, Scombromoris maculatus, in the Gulf of Mexico, but didn't have enough biological information on the species from that region. They considered using the abundant information in the literature for a population of Spanish mackerel from Brazil. But my colleagues and I showed that that population was a different, undescribed species, which grows a little larger, lives a little longer, and matures a little later. So it would have been a bad idea to use the Brazilian information to try and manage our Gulf Spanish mackerel. Let's use the account of that Spanish mackerel, the Serra Spanish mackerel, which we named Scombromoris brasiliensis, to show how we arrange the information on each species in our book. Each account starts with the common name, the scientific name, then Val's lovely figures, common names in French and Spanish as well as English, etymology or derivation, then field marks, and the field marks are illustrated with little inset drawings that Val created for this purpose. On the next page, we have unique features. We've got diagnostic characters, and the diagnostic characters include numbers of gill rakers, numbers of fin rays, vertebrae, color pattern, all the characters we use to sort the species out. Then geographic range, maximum size. And when we had the data, we have the IGFA all tackle angling records. That's the International Game Fish Association, which maintains records of maximum size for sport fish. Then the habitat, and over on the next page, reproduction, including size at maturity and numbers of eggs and things like that. There's the map of the distribution of the species. See, it doesn't extend up to the Gulf of Mexico, where it's replaced by a Gulf Spanish mackerel. Then fisheries interest, and over on the next page, threats to the species, conservation, and then finally, on the next page are a few of the most significant references for the species. My work shifted from taxonomy to conservation when Kent Carpenter at Old Dominion University started a campaign to add marine species to the red list of endangered species because previous work had been largely restricted to terrestrial and freshwater species. Kent asked me to form and lead a new IUCN Tuna and Billfish specialist group. We held meetings of concerned fishery people for several years in several different countries, as you can see in this slide. We published analyses of the threat status to each of the 61 species of tunas, mackerels, and billfishes on the 2010 red list of IUCN and summarized the result in a 2011 paper in Science. 7 species, that's 11% of the totals of 61, met the threshold for a threatened category. 2 bluefin species, and later we added the 3rd species, big-eyed tuna, blue and white marlin, and 2 species of Spanish mackerels. Our book is now being used as the basis for preparing a 10-year reanalysis of the threat status of all the tunas and billfishes for IUCN. And now I'll turn the program over to John Graves to talk about billfishes.
Thank you, Bruce, and good evening, everybody. What I'm going to do, Bruce has talked a little bit about the scombrids, and what I'd like to do is talk a little bit about the billfishes. But before I start that, I would like to note that I've been working, well, I first met Bruce Collette in the late 1970s when I was a graduate student at Scripps Institution of Oceanography, and Bruce would come to Scripps to look at the Scripps fish collection for some of his studies. So when I came out to VIMS in 1990 and started my program, Bruce was just a few hours away, and he has served on 8 of my graduate students' thesis or dissertation committees. And when Bruce actually asked me to be a member of the IUCN Tuna and Billfish Specialist Group, I jumped at the chance to do that. And as Bruce mentioned, when we did that 2010 threat status, we had 4 global meetings and then 1 where we put the regional meetings all together. So it was a great process and set up this book. And we had thought the idea, well, we have a lot of this information, let's go and turn it into a book. And Bruce said he thought he might be able to get Val Kells, and I was very familiar with Val's work, and that was like a home run right there. And we thought it'd be a 2 or 3 year project, but it took much longer than that. So what I'd like to do is just give a few insights here on the billfishes. And so Bruce looked at some of the species level changes that made the FAO book on tunas out of date. Here are some of the ones that made the billfish book out of date. So they recognized 2 species of blue marlin, one in the Atlantic, one in the Indo-Pacific. Genetically, those are actually the same species. Similar situation for sailfish. Also, Nakamura had 2 sort of what he called that were iffy species. One was a roundscale spearfish, and that was subsequently validated in 2006 as being a valid species. And then the hatchet marlin, which was even more iffy than the roundscale spearfish. And I'll talk more about both of these. The hatchet marlin is not considered to be a valid species, and I'll explain why just a bit. So these were the changes at the species level, but we also had some changes, higher order changes, and we actually changed the number of genera in a paper with Bruce, Jan McDowell, and myself, and we went from 3 genera of billfishes within the family Chapteridae to 5. And so while blue marlin and black marlin are morphologically very similar, it turns out genetically they're very different. And we ended up recognizing 5 different species, 5 different genera for the species here. So Bruce has already gone through what a typical species account looks like, and so we were each going to pick a species. The species that I picked is a roundscale spearfish for a couple of reasons. One, because we were instrumental in helping to validate it as a species, but also it's an important one here along the mid-Atlantic, and one that's been essentially unknown for some time. So rather than going step by step through it, I just want to do some of the highlights. So it turns out that the roundscale spearfish was described by Richard Lowe in 1840. And to put this in a temporal context, the white marlin was only described in 1860. So the roundscale spearfish, when it was described in 1840, was represented by a single specimen that was collected off the island of Madeira. And the person who brought it to Lowe's attention was Mr. George Butler Leacock, and hence the name Georgii. So turns out that Mr. Butler's great assistance to rendered in the cause of ichthyology was just making the specimen available. Unfortunately, that original specimen or the holotype no longer exists, and doubly unfortunately, Richard Lowe and many of his manuscripts and notes were lost in an 1874 shipwreck. So that made the description very difficult and problematic. And in fact, in 1960, Robinson de Silva, after studying the billfishes, said that the mystery of the roundscale spearfish will probably never be solved. Well, Robbins didn't give up. He went over into the Eastern Atlantic and studied billfish for some time. And in 1974, he published a paper suggesting that the roundscale spearfish was in fact a valid species, but that it was restricted to the far Eastern Pacific. And he provided some morphological characters to distinguish it. Fast forward to the 1990s, when I started my program here at VIMS, one of the things that I was looking at was population structuring within billfishes, and I was looking at the white marlin. So a good opportunity to collect genetic samples of white marlin is at the recreational tournaments here along the coast. And as I would be sampling at these tournaments, several anglers would come up to me and ask me, well, what's going on with this hatchet marlin? And so I learned that the story of the hatchet marlin, and it's simply, they thought it was a funny looking white marlin that had truncated or cut off dorsal and anal fins. And so I was on the lookout for that. And actually for the first 10 years of sampling that I did, I found very few fish that fit that morphological characters. But then starting in 2000, we ran into quite a few of them. And as we got more and more samples and genetic samples, we realized that what we were calling white marlin actually represented at least 2 different species of fish. And we published on that, and at the same time and at the same conference in the paper following us, Mahmoud Shivjay and his group down in Florida studying samples of white marlin and other species from the longline fishery taken by observers, realized there were 2 species, and they tied this mystery species back to that original description of the roundscale spearfish. So the roundscale spearfish was validated in 2006. Now it turns out that this truncated morphology, or this hatchet morphology, all roundscale spearfish that I've seen, and I've seen over 100 of them, have that morphology. But white marlin can have a rounded fin, but there are also some white marlin that will also have this truncated dorsal or anal fin. So the hatchet marlin is really just a physical morphological description, and that fish can either be a white marlin or a roundscale spearfish. But it's striking that the roundscale spearfish more closely resembles a white marlin than it does the other 3 species of spearfish. And here's just an example of 2 taken from a tournament. And on the top we have a white marlin, and on the bottom we have a roundscale spearfish. And using very nice keys that Val put in for the field marks, one of the things you can't see here is sort of the truncated fin, but also there's a difference in that a roundscale spearfish does not have spots on its dorsal fin, whereas a white marlin does. And both the dorsal and anal fin on a roundscale spearfish is going to be truncated. And again, some white marlin will have rounded fins, some will have truncated fins. But the easiest way to tell the 2 species apart is to turn them over and to look at the position of the vent. And it turns out that in a white marlin and in a striped marlin, the vent is just a few inches in front of the origin of the anal fin. Whereas in all spearfishes, the vent is much further forward. It's 6-7 inches in most specimens. And so it's more mid-body than it is close to the insertion of the anal fin. And that's a dead easy character. There are also differences in the length of the branchiosticles. These rays here underneath the mouth cavity here in roundscale spearfish, they extend to the edge of the operculum or the gill plate, whereas in a white marlin, they're shorter. There are also differences in scale patterning and also a little bit in the coloration, which you're not going to see from these dead fish. But fortunately, as Val will explain, she doesn't use dead fish pictures for her artwork. And so, to see a picture of a live roundscale spearfish is something else. They're just absolutely beautiful. They light up with this bright neon blue coloration, and they get it in different parts, and they can also get these feeding bars. And one of the distinctions that you can see between a white marlin and a roundscale spearfish is actually the formation of these bars, because in a white marlin, these bars will extend all the way down as single bars. But if you look at a roundscale spearfish, they actually bifurcate, many of them, alternating bifurcated, so they're like upside down Ys. You look at this little animal here, you can see that it's lost its bill. That's not a terrible incident for a billfish, because they can feed, they feed after that just like a tuna. They don't have their bill to stun prey, but they do quite well. And in fact, I have seen a higher proportion of roundscale spearfish missing their top or upper bill than I have in any other billfish species. And having been on a boat where we've caught some of these, they sometimes will come and attack the boat. And as Ken Neal, a person who's provided many opportunities for me and my students to get out to sea to do our work, he calls them one mean fish, and indeed they are. So there's not a lot of information known about the roundscale spearfish, because if you think about it, it was only really definitely recognized 15 years ago. We do know the range, though, extends much further out into the Atlantic than Nakamura indicated in his FAO guide. We know that it occurs throughout the Atlantic, We're not so sure about what is occurring in the Mediterranean because we haven't genetically typed a lot of the animals that are in there, but we know there are other species of billfish in the Mediterranean. But we do know the roundscale spearfish occur throughout the North and the South Atlantic. In terms of size, the maximum size that these fish get, the world record was about 73 pounds or so at first, and now it's and here's a tournament from just this August up in Cape May, New Jersey. Here's a 74-pound roundscale spearfish. They caught one 77 pounds in that tournament. They have been caught up to about over 85 pounds. And so as more people continue to fish for them and recognize that they're not a white marlin, but they're different species, I expect that the maximum size and the world record will increase. Movements, well, thousands and thousands of white marlin have been tagged by recreational anglers. But some of those white marlin were undoubtedly roundscale spearfish, but we don't know which was which. And that's a problem with all of the fisheries data for this species. But we do, our program, we tagged one roundscale spearfish with the pop-up satellite archival tag. This was my student, Emily Luce. She was hoping to look at differential habitat utilization by white marlin and roundscale spearfish and was planning to tag one of each species, tag a white marlin, then a roundscale spearfish, then a white marlin, then a roundscale spearfish, so they'd all be out for the same time and experiencing the same conditions. But she got her white marlin out the first day, and it took, I think she passed 110 fish before she got her first roundscale spearfish. And this animal was tagged off at Cape May, New Jersey. The tag stayed on for 34 days, and the animal pretty much made a beeline due east and about 850 miles before the tag popped off. Or another way of looking at it, in those 34 days, it was a third of the way to the Azores. Not much known about the habitat or ecology, but again, from the same one individual, so that's not a large sample size, we can see that the way they utilize the water column is very similar to both a white marlin and a blue marlin. So in the coastal waters, where we have a shallow thermocline, so it gets cooler at shallower depths, they tend to spend most of their time in the upper 50 meters, and in fact, most of their time very close to the surface. But then, and so we have the depth here on the left, and on the right, we have the temperature. And if we look at the temperature, you can see the animal's going along, and all of a sudden the surface temperature increases greatly. And that's when this animal, as it was moving east, crosses into the Gulf Stream. And when it hits the Gulf Stream, the water's warmer, and also the thermocline drops. So the habitat available for this fish is much greater. And you can see that it's spending a lot of time now, instead of between 40 meters in the surface, it's going down to 100 and dives even down past 150 meters. And so that's what we know, but not a lot. Food, we've looked at the stomach contents of a lot of these, and just like white marlin and blue marlin, they are opportunistic predators. If squid are available, they'll be eating squid. If small tunas, like the frigate tuna, are available, they'll be eating those. And year to year, at least when we're sampling at these tournaments, it will vary dramatically. Reproduction, again, we know a lot about the reproduction of white marlin, but we know next to nothing about the reproduction of roundscale spearfish. And again, an N of 1, but several years ago, I was sent a picture by a colleague in the Azores of asking if this fish was a roundscale spearfish. It was, and he also showed me a dissection of it, and it had a running ripe gonad. So they're spawning, at least in that area, in the month of May. Fisheries interest, obviously these all billfishes are taken as bycatch on pelagic longline gear. There are also some directed fisheries for billfishes. These are mainly artisanal or subsistence fisheries. And of course, there's the directed recreational fisheries. So there's quite a bit of interest for these animals. And the conservation status, well, it turns out we've been collecting records of white marlin and roundscale spearfish together. And in fact, in some areas, people have been misidentifying roundscale spearfish with the longbill spearfish. So there's a lot of problems with being able to sort those out. And essentially what they're doing now at ICAT, which is the management organization for the Atlantic, for the pelagic species, is they do a basket assessment. So they're considering all of these species together. The downside to that is if one species is responding differently than another, we won't know that. So that's one problem. And you think, well, could we go out and sample the ocean and figure out what the proportion of roundscale spearfish is to white marlin over there? And it turns out that probably won't work, because what we found out is that the proportion of white marlin and roundscale spearfish can change dramatically year to year and in areas. So just sampling off Cape May, New Jersey, as I sampled during the 1990s, out of 96 total specimens, 2 were roundscale spearfish, 94 were white marlin. Going through the next decade, we see that there's a higher incidence of roundscale spearfish, and in the last decade, it's already up. We're about 46.5% roundscale spearfish. So there's been a change in those, but again, this is very specific sampling. It's in one period of time. It's always in the 3rd week of August. It's within 120 nautical miles of the Cape May sea buoy, and it's taken with one type of gear. Furthermore, it's only the top 5%, the largest 5% of the animals that are brought in. And when we've worked with some other vessels that have fished throughout the year and gone ahead and identified every fish that they catch and release, it turns out that roundscale spearfish are not this abundant. They're abundant in a large size class in August, but throughout most of the summer months, you're looking at less than 5%. Typically of the white marlin would be actually roundscale spearfish. So in a nutshell, that's just a little of the backstory behind one of the species accounts. And I think we need to move on now to Val, because I think she's going to be really exciting because she really made the book with her illustrations.
Hi, everybody. My name is Val Kells. I'm a marine science illustrator. I fell in love with fishes a long, long time ago. My grandfather was a fisherman. He taught me how to fish. I went to the Florida Keys after my folks got sick of me running around on boats on Long Island Sound. And in a very long and winding way, I found my way to UC Santa Cruz, where I fell into the marine science illustration program. At the time, the science illustration program, where I was able to combine my absolute love for all sea life and my crazy need to illustrate nature as it appears and not in big wild colors. So this is where everything came to be. After graduation, I went on to illustrate for over 30 aquarium museums and nature centers in the United States and abroad. I also did extensive publishing in books, periodicals, and magazines. In 2011 and 2013, after I had joined forces with Johns Hopkins University Press, I went on to publish a field guide to coastal fishes from Maine to Texas and field guide to fishes of Chesapeake Bay. And as Candace noted, the first book, Coastal Fishes, Maine to Texas, has now become a staple in classrooms, on boats, and in research facilities. In 2016, I got an email where I published a field guide to coastal fishes from Alaska to California. This book took almost 5 years. And from there, I went on to publish Tunas and Billfishes of the World and Field Guide to Freshwater Fishes of Virginia. It's been an amazing ride, and I have loved every single minute of it. I also publish folding guides, posters, note cards, giclée prints. As a freelance illustrator, I have lots of fingers and lots of pies, and I've always got a million things going on. That's just the deal with being a freelance illustrator. So now I'm going to talk about the illustrations. So my job as a scientific illustrator is to illustrate the fish as it appears in life. There is very little room for fudging. I don't fudge. I take very, very close attention to morphology, to color, to placement, to all the diagnostics. There's no room to just make it up. I also rely heavily on photos of live fishes. As you can see, the top photo is of a dolphin fish that we just caught off of Ocracoke, and it's alive, and we let it go. And after they die, lots of fishes change colors, as in this fish down below here. So I try to illustrate all the fishes as they appear live, healthy, in hand at the top of the water column, which I'll explain in a few minutes. I also collect a lot of specimens. This is a mama-chog that we collected behind Ocracoke. We just pulled out of the water. It had been living in the grass, and after we put them in the bucket, they changed color. There's still both mamachogs, but I would never illustrate the mamachogs in the lower image because that's not how they look in nature. The photo is still helpful for fin placement, for the size of the eye and other morphological features, but the color is no use to me. As everybody knows, lots of fishes are moving, they're hiding, they don't ever face left with all their fins erect. So my job is also to illustrate the fishes facing left, which is a scientific norm, life colors with all the fins up, and yeah, it's challenging. Fishes also change shape after they die. This is my son holding a big eye that a friend of mine had caught and put in a cooler for me because he thought that I would love it, and I did. And although I would never illustrate this fish in this position, I could still glean a lot of information from this photograph. The other thing that I've got to deal with is the lack of color underwater. As most of you may know, as you get deeper in the water, light changes. Red goes away first, then orange, then yellow, and what's left is blue. So a red fish down at 250 feet would have lost all its red. So I illustrate the fishes as they would appear at the surface of the water. This is a part of my library. When I start my research, it used to be I'd have to go to a real library back before email and back before all this technology and check out the books, and now I've got a fantastic library of my own. And so I dig into this library, but I also dig heavily into the internet, which is just an enormous reference for me. I have a big library of my own photos. I am blessed and cursed with a photographic memory. That every fish that I've seen, I remember it. Even if I haven't caught it myself, I remember it. I take photos of all kinds of anatomy. I actually use that photo of the sheep's head jaw in the Field Guide to Fishes of Chesapeake Bay book. I take close-ups of their fins, close-ups of their scales. I never know when these photos are going to come in handy, and they often do. I also take photos of juveniles. These were all sane behind Ocracoke, and it's one of the best ways to get really good information about the juveniles is to get in the mud and pull them out with a net. Despite what John said, yeah, I do take pictures and use images of dead fishes. They might not be useful for their color, but I can get a lot of information about their morphology. So I do visit fish houses. I also use video. This is a screenshot of the rosy lip sculpin I found online. There were no images of this fish alive, but I had this live video, and so I actually illustrated this fish for Field Guide to Coastal Fishes from Alaska to California by hitting pause, play, pause, play, pause, play. It was pretty funny. When I hit a brick wall, I often tap into my co-authors and their extensive tree of associates. I've found that nearly everyone in the field is very willing and able to share their expertise, their knowledge, and their connections. And very rarely do I go empty-handed. So now I'm going to talk about illustrating the fishes. This is what my desk, my table, my drafting table looks like on any given day. On this day, I was illustrating a starfish for a folding guide of the Pacific Northwest. You can see I've got my drafting tape and I've got my laptop, my palette. Yes, I use toilet paper. The toilet paper is a secret weapon. I use watercolor and gouache almost exclusively, and when I make an error, the toilet paper comes out and bam, error fixed. This is a palette that I've been using since I was in college, and I can create any color under the sun with this palette. I am not hindered. A lot of people think that watercolor is very challenging, but after using watercolor for 36 years, I'm a master of it, and I love it. Okay, I'm going to talk about the baby sailfish. So I'm deep into the Tunas and Billfish book with John and Bruce, and I wanted to add a lot of juveniles because they're morphologically different from the adults. most fishes, virtually all fishes, change shape, size, color as they mature from larva to juvenile to adult. And we had the room, we had the time, and so I said, yeah, let's add the juveniles. So we added juveniles wherever we possibly could. And there were lots of photographs of the juvenile sailfish to choose from. I had live images, I had dead images. You can see this one at the lower right from the Australia Museum, which was particularly helpful because it showed the fish flat with the fins up. I could use it for measurements. So I was able to put together the preliminary drawing, which is the first thing that I do. I do the preliminary drawing in pencil on tracing paper, and I can erase and change and move things around. I put a grid over it until I'm happy with the final product. The way that I see the preliminary drawing is as a blueprint. If the preliminary drawing is wrong, then the illustration is going to be wrong. If a blueprint is wrong, the building is going to be wrong. So I take a lot of time and care in the preliminary drawing. And here I am getting ready to transfer the preliminary drawing to my watercolor paper. I use a 300-pound hot press, which allows me to add a lot of water and a lot of washes to the surface, and the paper doesn't buckle. You can see my array of paintbrushes here. I have lots and lots of paintbrushes. I go through them on a monthly basis. I literally wear them out and retire them. And each one either gets repurposed or retired, and they all have a special use. I actually have a special paintbrush just for the irises of the fishes. So here I have, I've used a graphite pencil to carbon up the back of the preliminary drawing. I've taped it down to the surface, and then I use a very hard 9H pencil to trace the graphite onto the watercolor ground. I then lift the preliminary off, and what's left is a very, very fine outline of the preliminary drawing in pencil. I use a really, really sharp number 6 sable brush to trace over the pencil lines. And then I erase the pencil with a very soft eraser. This is one of my other special weapons, a click bic or a bit click. It's very soft, it doesn't harm the surface of the paper. And then once this is done, I just start painting. I turn on my music, I go into a Zen mode. I already know what the fish looks like. And I've got all my images up on my laptop. I've got images up in books. I've memorized it, and I just start to paint. I start layering the watercolor, and I keep layering and layering. And then about, I don't know, this took about 6 hours maybe. About 6 hours later, it's done. But even now, I'm not done. I scan the image, and then I save it, and then I import it into Photoshop, where I have to delete the background from the image so that I have a silhouetted image. And this allows me to superimpose image on image. It allows me to add a drop shadow. And then once all this is done, I import it into the book design. As Bruce mentioned, he and John worked on the text. They sent me the text. I dropped the text into the book, and then I start adding the illustrations and working things around, adding the maps, adding graphics. And this is what the final pages look like with the illustrations, the text, the maps, and there's that little baby sailfish. So this is not the normal way that books are usually produced. Books are usually produced, and the authors do their authoring thing, the illustrator does their illustrating thing, the photographers do their photography, the map maker makes the maps, and all that goes through the copier, then back to the authors, back to the copier, then to a designer, and the designer designs the book, and they spit out the book, and the authors, the illustrator have no idea what the book is going to look like. But since Johns Hopkins had already published 3 of my previous books, 2 of which I had already designed, they had confidence that I would be able to also design this book. So they said, go for it, Val. And this gave me and John and Bruce control over the book. So our process looks like this. Bruce and John did their thing. I did my thing. We got the photos that I needed to get. I used the photos for references. We had our maps done by another map maker. Everything was given to me as a designer. The whole thing went to our copy editor, Debbie Bors. I went through extensive copy editing. I think it was 5 or 6 rounds of copy editing. Back to me to redesign it. Back to the publisher. I don't have that step in here. Back to the publisher for peer review. And back to us. Then back to Hopkins. Then it is finally uploaded to our printer in Japan. And then we all keep our fingers crossed that it makes its way safely back over here to the United States and into the warehouses. This is an image, I forgot about this one. This is an image of what copy editing actually looks like. It's very time consuming, and bless Debbie Bors, she did an excellent job. So anyway, so after we get through all the copy editing, all the printing, and all the shipping, the books finally arrive and the fun begins. And here's my box of books that arrived here just about a year and a few months ago. And there's the book with my palette. So with that, I'm gonna hand it back over to Candace, and we can take some questions.
Awesome. Thank you so much, everyone. That was so great. All right, so we do have some questions that are coming through. The first one is for Val. Have you ever illustrated larval fishes?
Yes. There are a few larval fishes in the Tunas and Billfishes book. If I just had to illustrate larval fishes for the rest of my life, I would be the happiest camper in the world. I just think they are the coolest things. So yes, when larval fishes come across my desk, I'm illustrating larval fishes.
Awesome. So Lauren Ford asked if you had to make any revisions to your book before it got published, and I think you sort of answered that briefly, but could you go over like any revisions? Like, is there anything that sort of differentiates this sort of revisions from a normal book revision?
Well, I don't know about Bruce or John, but our book is so recent, we're not aware of any recent revisions. When Maine to Texas came out, the powers that be decided that groupers needed to be separated from sea basses. In that book, all the groupers and sea basses were together. And now recently, the powers that be have decided that parrotfishes and wrasses need to be in the same family instead of 2 families. So those kinds of changes do warrant a major revision. So down the road, I am going to be working on the 2nd edition of Maine to Texas, and further on down the road, another revision of Alaska to California. But as far as I know, we don't have any major revisions to this book because it is so recent. I defer to Bruce and John.
Taxonomically, that there aren't any revisions at this time. There may be in the future, but there's certainly the biology of the animals. Papers are coming out all the time. And so many of the accounts, you know, there might be minor updates to them. One that's sort of significant for fisheries is that the maximum age of yellowfin tuna was extended from 12 years to 18 years, which has a large impact on the assessment of the tunas, but it was just, that came out just a few months ago. So, you know, we're keeping track of that information, and if there ever is an update, we'll put it in there. And Bruce, like he said, has an incredible bibliography of tuna and billfish publications.
Perfect. That sort of answered another question that came in from Julian Pepperell. If you think the science is settled on the taxonomy of billfishes, or if it's possible that future revisions may be necessary, like possibility of sailfish subspecies.
Sure. Want me to take that, Bruce? I'll take it. So, hey, Julian, good on you. Julian's coming in from Australia, but there's always a possibility. You know, a lot of our earlier genetic studies were done with limited number of molecular markers, and now we're going up to using thousands with next generation sequencing. Your specific question for the sailfish, we do have next generation sequence information on the sailfish, and we see nothing that indicates subspecies of the sailfish. There is some differences in the mitochondrial genome, but that's a single locus that's maternally inherited, so differences of isolation will not be swamped out through recombination. So you're seeing historical separation there, but I would never say that anything is set, you know, but and eventually we will have next generation information for all of the billfishes, and that at that time I would think that we're going to be pretty set on the taxonomy.
And on that note, we do have another question. We have one from Cynthia Edwards. Do you believe that there are still tunas and billfishes that have not been seen and or identified?
There have been attempts to describe new species, but so far, most of them have not been valid. That doesn't mean there aren't any. And part of the changes we made were not new species, they were splitting populations. The two Spanish mackerels were quite a surprise because they were both popular sport and commercial fishes. They just were mixed up with other fishes. So maybe, but I haven't seen any evidence recently.
What are some of the priorities and challenges for management of billfish, which are not necessarily commercially important, but recreationally important? And what future research is needed to support future management? That question comes from Laura Smith. So there's a lot in that. So first of all, what are some of the priorities and challenges for management that are recreationally important for billfishes?
Well, in terms of managing them, we have very poor landings information for billfishes. We also have problems with species identification. Many countries that report their billfish catch don't report it down to species. On a larger picture, it's true that billfish are not, individually very expensive, they're not a high-priced item. And so they're not the target species for most large-scale fisheries, but they are kept, or at least they interact with the pelagic longline gear. And so you have a situation where the stock status for the billfishes, they're overfished. Their populations are lower than we'd like them to be, but some of the target species that the pelagic longliners are fishing for are healthy. So we're trying to find ways to minimize the interaction of billfish with the gear, or if there is interaction, to increase the likelihood that these fish can be released alive. And that's difficulty. I mean, and the same thing in the recreational fishery. Many individuals go through great pains to make sure that their fish are released alive. And even here in the US, the recreational anglers switched over from J hooks to circle hooks in natural baits, simply because when they found out that J hooks resulted in a fairly high post-release mortality rate compared to circle hooks.
Wow, okay, so that's a lot that we can link into. That's awesome. So this question is for Bruce, also through Julian. How often does the IUCN specialist group meet and or review information?
What we're doing now is I have a small group of specialists that are reviewing stock assessments for all the major tuna species that are commercial species, the bluefins, yellowfin, big eye, and skipjack. And we've had one preliminary meeting with half a dozen specialists to review our progress. So the committee is the committee that formulated all the red list things was a sort of floating committee. We had a number of people that were at all the meetings. And then when we moved to other countries, we had a lot of local people. So the total number of people that were reviewing some of the species were dozens or scores or maybe as many as 50. So we don't have regular meetings. What we're doing now is taking all the data that's in the book and recasting that in the format of the red list. And then we're having people review segments of it. And then the next step will be to send the reviewed manuscript out to all the people that reviewed it originally for the 2010 to see if they have any comments.
We do have another question about the billfishes. What do you mean they attack the boat?
Well, specifically, you get the fish near the boat, and rather than swimming away, it just swims straight at the boat, and the bill hits the boat. And this is not it's not just roundscale spearfish that do that. It happens quite a lot in other areas. In fact, some of the older wooden boats fishing out of Kona in Hawaii, the blue marlin would charge them, and they would actually, the bills would penetrate the hull of the boat, and the animal would break it off. But they would then just cut off the bill flush with the hole, seal it, and that was a mark of pride on the boat. Some of them had several billfish bills stuck in the hole.
Oh, wow. That's really interesting. And then, Val, we have a question for you. Someone noticed that the fishes on the cover of one of the books all face to the right, the one about the Pacific, and they were just curious about why that is and how did that happen? Since it's typically faced to the left, is there necessarily a reason behind that?
Yeah, the scientific norm for fishes is to all face left. And I'm just going to go into a little background about the covers of the book. I fought long and hard for the covers of these two books. The design department wanted a cover that was very typical of all other field guides, with a mishmash of fishes, all different sizes going, and it's just like, they all look the same. I really wanted something that looked different, that was going to stand out. And so I fought for the NOAA charts. You'll notice there's a NOAA chart on Maine to Texas as a background. And that NOAA chart is of Ocracoke, one of my favorite places in the whole world. And it has virtually every habitat on the East Coast, except for Rocky Coast. Obviously, it's got open ocean, it's got estuary, it's got inlet, it's got beaches, it's got bay. And then with the fishes, we decided to have them facing right to also have the cover stand out, and to also say, hey, everybody, kind of open the page. Let's open the page and see what's inside. So that's the history behind the covers.
We have a question coming in from George Sudbury. Many of these species require international cooperation for management and conservation. How is that achieved, and has there been any improvements over recent decades?
George, as you're probably aware, you have your 5 tuna regional fishery management organizations, and so you have countries that are working together, adopting measures, but then it's incumbent upon each country to enforce those measures, to report their data, and so you're hoping that everyone's going to play fair. And if they're not, the group as a whole can do multilateral sanctions. Some of the most strict being actual trade measures, but it usually doesn't have to get to that. There's a lot of peer pressure in these international fora, and I think that you're seeing more and more countries working together. In the Atlantic, there's been several, of these multinational stocks that have been rebuilt, whether it's North Atlantic swordfish, North Atlantic albacore. They've all improved. But the more players that you have fishing on a particular stock, and the more different types of fisheries, the more difficult it becomes. And in the case of big-eye tuna in the Atlantic, where they're catching the very small ones in a purseine fishery for skipjack, and yet you have your directed longline fishery for the adults. And different countries have these fisheries. It's a mess, and absurdly, they manage using consensus. They rarely vote. And in the case of ICAT, you have 52 member nations, one of those being the EU, which represents 27 nations. So it's a mini UN, and the idea that you're going to get complete consensus on these measures it's a challenge, put it that way.
John should know, since he's been chairman of the advisory committee for ICAT for eons.
We have time for a couple more questions. Val, are there any fishes that you find are more difficult to illustrate compared to others?
Oh gosh. The most difficult fishes to illustrate are those that I have the least amount of reference for. Yeah.
Like we never got a good reference for one Scombromoris.
Do we finally have a good reference for the plain bonito? We had a few dead fishes and not a whole lot else. We had people picking across the Mediterranean for specimens for me, and I eventually just had to Frankenstein the thing, knowing what I know about tunas. The fishes that are the easiest are the ones where I have scads and scads of information. I can illustrate any fish under the sun. I am not intimidated. So yeah, the most difficult ones are the ones that I don't have a lot of information. If I've got the information, I can pull it off. That's it.
That's awesome. All right, we have quite a lot of questions here, so I'm just going to go with one last one. So for our last question, what are your next projects going to be?
Well, I can go first. After I finished Tunas and Billfishes, and after I was in the middle of Tunas and Billfishes, I was in the middle of Virginia Freshwater Fishes. I'd already signed on with Hopkins to illustrate, co-author, and design the third in the series of Field Guides to Coastal Fishes. So I'm right in the middle of Field Guides to Coastal Fishes, Bermuda, Bahamas, and Caribbean Sea. I'm wading my way now through gobies. Yeah, the third most populous family of fishes. It's going to be interesting, but thankfully most of them are well documented, and I've got great people to tap into. Thanks, Luke. So that's what I'm working on. And after that, we are going to dive into marine fishes of North America. And that's still a TBD, but I have a thumbs up from my editor at Hopkins. More than likely, it's going to be a multi-volume book, and it might be my mea culpa. I don't know. I've got friends who say, "No, Val, you're just going to keep going." So that's what I'm working on now and next.
My next project is to finish the reevaluation of all 61 species of tunas, mackerels, and billfishes for the IUCN Red List. And we've got drafts of the first 7 or 8 species, but we've got a long way to go.
And Bruce is getting me those drafts, so I'm in the IUCN process as well. But and I don't see any books in the near future for me, but lots and lots of papers to do before I retire at the end of next year. Although when I retire, I don't anticipate I'll be going anywhere. I'll just not be getting paid for what I'm doing and like to do.
Very cool. Well, thank you all so much for joining us, and thank you to all of the attendees, and thank you to our generous supporters for allowing this to happen. And we hope to see you all next year at our January After Hours. Thank you so much and have a great night.